Good evening, everyone, or good morning, depending on where you're joining us for this seminar. Today is another exciting day, and we get to listen to two speakers lined up today. But before we start this seminar, I would like to do the land acknowledgement. Iowa State University aspires to be the best land grant university at creating a welcoming and inclusive environment where diverse individuals can succeed and thrive. As a land grant institution, we are committed to the caretaking of this land and would like to begin this event by acknowledging those who have previously taken care of the land on which we gather. Before this land became Iowa State University, it was the ancestral lands of the Bakoje or Iowa Nation. The United States obtained this land from the Meskwaki and Sauk nations in the Treaty of 1842. We wish to recognize our obligations to this land and to the people who took care of it, as well as the 17,000 native people who live in Iowa today. Again, it's a special occasion. We have two speakers lined up, but who are they? I'm going to introduce them. Leonor is a professor in the Department of Plant Pathology and Microbiology at Iowa State University. Her research focuses on the biology and epidemiology of fungal diseases of soybean, with primary interests on soilborne fungi that cause soybean sudden death syndrome and soybean root rot. She has conducted research on the impact of diversified cropping systems on soybean health and productivity since 2010. Some of her current research focuses on how cover crops and biochar amendments affect soybean diseases. She also teaches courses in mycology, research ethics, and professional speaking skills at ISU. Our second speaker, Larry, is an associate professor in the plant pathology and microbiology at Iowa State University. His research focuses on identifying and understanding molecular, cellular, and ecological mechanisms contributing to bacteria, microbe, and bacteria-plant interactions. Past research has elucidated how bacterial water deprivation tolerance and biofilm formation mechanisms are intertwined. Constructed synthetic biology tools, e.g. the life history biosensors, and showed that bacterial vitamin B12 production stimulates algal thermotolerance. More recently, his lab is using CRISPR interference to identify gene networks required for microbiome assembly in the maize rhizosphere, developing aptamer-based imaging tools for microbial secreted public goods, using synthetic microbial communities to reveal bacteria-bacteria interactions, modulating microbiome assembly and maize nitrogen use efficiency, characterizing microbiomes and their metabolic functions of high nitrogen use efficiency maize, and the contribution of soil and root-associated microbial communities to diversified cropping system benefits. It is an honor and pleasure to listen to these two speakers, and at this juncture, let's welcome them.
Thank you for the introduction, and it's a pleasure to be here, and thanks, Marshall, for giving us the opportunity to talk about our project here today. So as was already mentioned, this will be myself and Larry presenting this talk. But before I start, I'd like to acknowledge also other collaborators that have worked with us on this project, primarily Dr. Matt Liebman, who's an agronomist. And without him, we couldn't have any of the data we're going to show, because he established the original field trial where we collected the data and worked with us through the whole project. Gwen Beatty from Plant Pathology, who has worked with us on also the microbiome and overall objectives of the project. Phil Dixon from Statistics, Andrew Hans, who's a ag economics staff who helped us with the economics analysis, and then the three students on the bottom were grad students that worked with us on the project. And also wanted to acknowledge our funding. So a lot of this project was funded by the USDA NIFA, also by the Iowa Soybean Association and Leopold Funds, among other things. So we'll get started. So as we mentioned, this talk is divided into two parts. The first part is what I'm going to present. I'm going to describe the experimental design for this trial, talk about the disease and yield data we collected, and also just very briefly touch on the economics of this, this experiment. And then Larry is going to take over and talk about the root and soil microbiome, and also about other soil parameters that were measured and how they associate with the data we collected. All right, so starting off with where this trial took place. So the research was conducted at the ISU Marsden Farm. This is located in Boone County, just south of the agronomy farm. And Matt Liebman started this trial in 2001. So it's been around for what, 20 some years, 21 years. His goal was to compare different cropping systems. There's 3 different cropping systems, and look at how those different systems affected plant productivity, the economics, and also weed management. So he had this trial going on for all these years. You can see here's an aerial photo of that location, and you can see there's 4 blocks. And then within each block, there are 9 plots, and the plots are very large. They're 83 meters by 36 meters wide. And there's a total of 9 plots in each block, because we have 3 cropping systems. And as you'll see, there's 2-year, 3-year, and a 4-year cropping system. So each stage of the rotations are represented every year. Okay, so large trial. And these, yeah, we'll talk about the rotation. So what are the treatments that were in this location? The first treatment is what we call the 2-year system, and that's your conventional corn soybean rotation. I like to call it an alternation, because you're really not rotating. You're alternating corn and soybean, and you're using synthetic fertilizers. So it's very conventional management, the system that's prevalent here in Iowa and most of the Midwest region for soybean and corn. There's a 3-year rotation system or cropping system that has corn followed by soybean. And then in the 3rd year, there's oats intercropped with the red clover. And so then it keeps going back to corn, and every 3 years it'd be back on corn or soybean. And then the fertilization happened primarily in the form of composted manure. And then there was a small amendment in a synthetic fertilizer as needed. Usually, I think this reduced fertilization from synthetic fertilizers by about 90%, between 80 and 90%. Okay, and then the 4th year system was a 4-year rotation, corn, soybean, and then oats intercrop with alfalfa. And then alfalfa would stay along and do a 4th year of just alfalfa. And then again, fertilization was similar to the 3-year rotation. Some of the tillage practice was slightly different, especially because of the oats and red clover. You needed to do some incorporation into the ground. So tillage varied a little bit. So this is truly, these are cropping systems. They're not just a rotation. You have fertility management and tillage. That's a little different. So the trial, like I said, was established 2001, has been very healthy. In 2010, it was a very wet year. I don't know how many of you remember that we had a summer that was a lot of rainfall, a lot of floods, a lot of moisture. And that year, Matt contacted myself and a colleague, Allison Robertson from plant pathology, said, I have this disease in my plots, and it seems to be affecting the different treatments differently. So we went to the field, and this is a picture of what we saw. So these are 2 soybean plots side by side. One of the plots looks pretty green, pretty healthy. And the other plot is very poor, has a very high severity of a disease, right? You see defoliation, the plants are yellowing. And so when we looked closer, we determined what disease it was. And one thing I should mention is that in every block that we went to, these large plots, every block had the same kind of pattern. And so this picture here shows you the 2-year system. So the corn soybean system was very heavily affected by this disease. And the 3-year system, the plants looked pretty healthy. There was some disease, but we're pretty healthy. The same was true for the 4th year rotation. And like I mentioned, every block we went to, this same pattern was repeated itself. So when we inspected the plants more closely, we saw that they showed some interveinal chlorosis and necrosis. So it's death or yellowing of the leaves between the veins. And a lot of the plants in the more severe patches had defoliation. So premature defoliation where the leaves fall off, but the petioles stay attached to the plant. Does anybody in the audience want to take a guess of what disease this might be? Does it look familiar at all? No? All right. It is called sudden death syndrome or soybean sudden death syndrome. Usually we refer to it as SDS. And so I'm going to give you just a little history on or background on this disease. It's the disease I primarily work on in my research program. So it's caused by a fungal pathogen called Fusarium virguliforme. It's a mouthful, so we often call it FV just because it's easier. It's a fungus that produces macroconidia. I am going to use the pointer here. Macroconidia are asexual spores that are kind of banana shaped or canoe shaped. And they're produced in large amounts in soil, on plant material. It also produces chlamydospores, which is in this yellow image. And those are also asexual spores, but they're round, they have a very thick wall. So they're dormant spores or survival spores. And those spores can live in soil for decades. So you may not plant soybean for 10 years or 15 years. And if you plant soybean again, they can germinate and infect, at least a large majority of them. So they're very resilient spores in the soil. So the fungus survives in soil, survives in crop residue. So roots that were infected, the plant material that may harbor the fungus. And then also it's been found in the soybean cyst nematode cyst. So there's an association between the two. And it spreads through movement of soil. So if you have equipment, machinery, anything, your boots, you go into a plot that has a fungus, you can take it to another part of the soil. The fungus infects the roots and stays at the root level. It never climbs up the plant, but it produces a toxin that causes the leaf symptoms. So here in this image, you have a cross section of a diseased plant, an infected plant with the red circle. And you can see there's some discoloration, right? The root cortex is kind of brownish. And beside it is a healthy plant. So the cortex is very white. And so the roots have poor development. And also, if the soil is moist, if there's enough moisture, the fungus will come out and sporulate on the root surface. And so these blue masses over here are basically the macroconidia, a lot of spores just forming attached to those roots. So imagine if these stay in the soil, you're just going to be adding to that inoculum inside the soil. The problem with this disease is that it's very damaging to yield. If the conditions are favorable for disease development, there's enough moisture, if there's enough pathogen, some fields will have up to 100% yield loss, and at least some parts of the field. So in the United States alone, you can have up to nearly 2 million metric tons of loss in a year, $750 million per year. So depending on crop prices and all this, but very, very damaging. And also in the map, you can see that it's widespread in the US. Pretty much everywhere that grows soybean in the US has this disease. Iowa, Illinois, Indiana, Minnesota, those are areas where it shows up a lot. So this was pretty concerning. It's a big problem in Iowa for our growers. So going back to the Marsden farm plots, so we're seeing this big effect, right? We're seeing that, as you can see in this data, the 2-year rotation, the corn soybean rotations had very high incidence. And so the graph on your left is incidence, so percentage of plants in the plots that had symptoms. And you can see there are 2 varieties. Variety 1 is the green one, is most susceptible. Nearly 100% of plants had this disease. The other variety was more resistant, and about maybe 30% of plants had the disease. But compared to the 3- and 4-year rotation, those same varieties, much lower, right? About 10% or less of the plants had disease. And when you look at severity, and that's how much of the canopy of a particular plant or the plants that were diseased is affected. So in a patch of disease, how much of the leaves are affected, the leaf area. And again, you see the same kind of pattern, much more severe disease in the 2-year rotation than the 3- and the 4-year rotation. And when we looked at yield, then we saw, compared to the 2-year rotation, so we had in the 2-year rotation up to 33 bushels per acre loss. Or the other way to say it is that in the longer rotations, we had 12 to 33 more bushels per acre than in the conventional system with the corn soybean. So really damaging on yield, and it was very highly correlated to yield loss and not other factors in the plots. So we thought, well, this is very interesting, but is it just something that happened this one year? Maybe something happened. So we decided, let's continue the study and look at it over multiple years. So what we did, this is a diagram of those plots. The ones that have the little yellow dots are the ones that are in a particular year in soybean, because remember, you have 2-year rotation. So you have a corn and a soybean plot in 2-year rotation. You have a corn, a soybean, alfalfa, and clover in another. So each year there's 3 plots that are in soybean. And so we decided to do a more systematic sampling. We developed what we call quadrats. And all those are is 3 meter by 1.5 meter rectangles, and are really squares, rectangles that are evenly spread. And so since the plots are so large, it allowed us to get to quadrat 1, rate disease, collect yield, and then as you'll see in a little while when Larry talks, also in some of these quadrants collect soil to get microbiome data. So we could connect all these different variables. And so it became a more systematic, more intense sampling. And so here's some disease data from 2010 to 2018. We did continue a little past this, but I think the patterns are pretty clear. This is actually disease index, which is a combination of severity and incidence. So it's severity multiplied by incidence and then divided by the severity rating. And so the story here is very clear that particularly in the years marked here with the red box, that we had high moisture, high rainfall, and it was most conducive for disease. You see much, much higher disease index in the 2-year corn soybean rotations than in the other longer rotations. And even in the other years that maybe weren't quite as favorable for disease, the trend is still there, right? You still tend to see a larger bar in the dark blue. So this showed us this wasn't just a 1-year thing. This was consistent. And remember that the plots change, right? 1 year, the soybean plot, the next year is another plot that's in the 2-year rotation and 3-year rotation. So this wasn't just those specific plots, it was the system itself that showed this enhanced disease. Okay, so what else did we collect? We collected yield data. This is for those same years, but only up to 2015. But yield loss, obviously also very, very significantly impacted. These are the 2 years that had high disease severity, and you can see that the yield loss is much greater in the 2-year rotation, and they yield much better in the longer rotations. Okay, the other thing we did at a very preliminary stage in these early in the first few years of the study was we had a molecular tool. We had a qPCR protocol that was supposed to be specific or was specific to Fusarium virguliforme. So for the 2-year and 4-year rotations only for corn and soybean plots, we collected soil, and Gwen Beatty and her lab isolated DNA and quantified the amount of the Fusarium virguliforme pathogen. So is this because we're seeing a buildup of the pathogen in the 2-year rotation systems than the other ones? And what you can see in this plot here, if you, for example, compare on the soybean side, the 2 and the 4-year, we do have significantly higher amount of FV DNA in the 2-year plots than in the 4-year plots. And interestingly, in the corn plots, we see the same. So this is not just in the soybean year. Those rotation plots, even when they're in corn, they show a higher amount of FV of the pathogen in the 2-year compared to the 4-year. So this indicated that something's going on that's reducing the amount of pathogen in the soil. It's affecting the disease, it's affecting yields. And so this led to myself and Larry and Gwen and the others collaborators I mentioned to put together a proposal to NIFA and saying, look, we have these observations and this data that shows there's a very strong effect of the cropping systems. We need to figure out why, what is happening and what can explain this reduction in SDS in the longer rotations or the longer systems. And so we proposed 3 objectives, and we're going to really talk about 2 today. The first objective, which is what Larry's going to talk about, we wanted to identify the key microbial and soil variables that would correlate to this SDS suppression, soil health, soybean productivity, that higher yield. So Larry's going to talk about the sampling we did at those quadrat levels. So we selected a few quadrats. There was microbiome analysis, different soil properties, soil physical and chemical properties. We continued to collect in my lab severity and incidence. The density in soil was also part of the microbiome study, and then yield. And then there's regressions and all different kinds of analysis that will probably take us 20 years to go through all the data. But just to see if what's correlated, what are the variables associated with this effect we're seeing. We had a second objective looking at with a hypothesis that maybe it was the oats, since oats are allelopathic plants. Is it an oat effect or is it the manure amendment? So we did some work there, but we don't have time to talk about that today. And then we also did an economic analysis where, thinking of the grower perspective, we were saying, well, okay, so these long rotations seem to suppress the disease, but there's also seed treatments available to suppress STS, to treat STS. So what would happen in terms of economics for a grower if we add the seed treatment in the 2-year rotation or the 3-year rotation or the 4-year rotation? Is it more economically viable to do a 4-year rotation without the seed treatment, or to pay the cost of the fungicide and do a 2-year rotation and just add that cost? So I'm going to just very briefly show you some data on the economic analysis, and then we'll hand it over to Larry. Okay, so this is for that study was done 2017 to 2019. Joey Booty was our student who collected a lot of this data. And so the first thing you can see on the graph on the left is, again, that pattern that they yield. And this shows 3 different years. So the little dots, I'm sorry, I forgot to put the label, but the red, blue, and green dots show the 3 different years. And you can see the pattern is very consistent. Typically on soybean yield was higher in the 4-year rotation than in the 2-year rotation. And the 3-year rotation tended to be in between, sometimes closer to the 4 years. So we do have higher yields of soybean. It doesn't happen every single year, but if they're not higher, they're pretty similar. So our goal was like, okay, but what inputs are going into the 2-year rotation, 3-year and 4-year? And at the end, the grower, what's the profit, right, that the growers get? And so to simplify a pretty detailed analysis, we looked at the cost of the inputs, we looked at the profit from selling the grain, and that gives you the return to land and management. So basically what money is left, what's the gain, right? And the 2 bars, you can see no flupiram. That's just the seeds are not treated with this fungicide against STS. And the flupiram, the yellow bars, are when you had the seed treatment. And so the story here goes, well, 2018, we didn't have a lot of disease. And so really there was not a lot of, actually, I think this year, what happened this year? We didn't have a lot of gains. I think it was a really bad soybean year. It was a drought year. It was a drought, that's right. It was a drought year, that's what I knew. And so just productivity wasn't great. But in the other 2 years, you can see that in addition to seeing you have more return as you get towards the 4-year rotations, you tend to have a greater return. We also see that when you add the flupiram, you tend to have less return. So the cost of the fungicide wasn't beneficial in terms of like protecting the soybeans from STS and protecting yield. So it was more cost effective to actually not apply the flupiram, even in the 2-year rotation. At least this year, that's not true in 2017. And in 2017, we did see a little, we saw basically the same pattern. So in this study, it's 3 years with not a huge amount of STS, but this suggests at least the benefit of the crop rotations, the longer rotations overwhelms is greater than the benefit of applying the seed treatment. Okay, at this point, you have the background, the stage, and Larry will continue and talk to you about the microbiome.
So at this point, I'm just actually going to pick up where we left off when we wrote our proposal. And this is directly from this. And we said, well, we propose that the soil and root-associated microbial communities are central drivers of many of the diversified cropping system benefits, and that really the knowledge of these communities is critical starting point. That's really all it is. It's a starting point to really reveal what the mechanisms may be. And this study was the second study that we've looked at the Marsden site. The first study was also funded by USDA and NIFA, looking at the maize plots. Okay, and so I'm going to share with you some of the information we'd be able to gather from looking at the diversified versus the conventional system. In that study, we only did the bigger contrast, the 4-year and the 2-year. But we are able to obtain some information that provided some insights to what might be going on here. First of all, using a particular method, we ascertained that there was more microbial biomass in the diversified systems. There was greater protease and glycosidase activity. So they secreted more extracellular enzymes to degrade the more complex polymers present in the diversified systems. We actually had, interesting, from my perspective, lower soil nitrate pool sizes in the diversified system, suggesting that there may have been less nitrification and or better nitrate uptake by the plants. Okay, we can't say what lower pool sizes. I like to think it's nitrification. In fact, we have data to suggest that. There were higher organic matter contents, not surprising because you added the composted manure. And then you also had, from our survey of bacteria, doing what we call amplicon sequencing. That's where we use ribosomal RNA gene signatures to get a profile of who's all there. We also do this with fungi as well. And that we could say that there's certain groups of microorganisms that are much more abundant in the diversified systems. And many of these organisms are known, particularly the Vericomicrobia, they're associated with more prairie systems. Okay, agriculture's made them basically disappear from agricultural lands. Prairies, they can be dominant, but once you start plowing, doing other things, they just disappear. Okay, and then also we observed that bacterial communities in the diversified cropping systems are much more interconnected. And so that laid the foundation of which we could then explore a little bit more about what's going on in terms of the microbial communities in these systems. Okay, and so I'm not going to go into a lot of the details of the molecular aspects of microbiome profiling. I'm just going to say is that we looked at bulk soil between the rows. We used the grid pattern that Leonor had established, but we could not afford to do every single quadrant. We could only afford to do 3 quadrants out of the 10 in any one plot. Okay, but that meant, though, with 4 blocks, we had actually 12 samples for each of the different rotations. Okay, and then we also looked at rhizosphere. Well, we couldn't look at the entire rhizosphere, it's too big. So what we decided is we were focused on something, because it's developmental stages. We would start at a developmental stage that we could get 5-centimeter root of the taproot. And then we did the 5-centimeter taproot segment for each developmental stage. Okay, and we removed the lateral root. So we only were looking at a small portion of it, the taproot. Okay, and only the top 5 centimeters of it at that. Then we took that, we washed everything off. That means there was soil attached to it and also bacteria attached to it. And that's what we call the rhizoplane, but we pooled them together as our rhizosphere sample. Those are physically interfaced with the soil and also the loose aggregates. Then we also took what we call the endosphere, what's inside the plant. We removed all the nodules, because otherwise our analyses would be all rhizobia that have dominate the nodules. So we had to remove all the nodules. But then we also trimmed the ends so that we didn't have any washates going in and contaminating the sample. And that would be what's in the plant, our endosphere. And so we did a lot of this. We did our field sampling. We did a lot of DNA extractions. We did quantitative PCR to measure the abundance of FV using the same set of primers that Leonor talked about earlier. We looked at total fungal, bacteria, archaea abundance, different microbial taxons. We did Illumina high throughput sequencing, and we look at the fungal community as well as the bacterial community. We ignored the archaea, which unfortunately happened. I would choose not to, but we just missed them in our way we did the analysis. Then we had a bioinformatic pipeline. It takes a lot of time, as Leonor said, a long time to look at. And there's a couple things that we did that are worthwhile pointing out. We did differential abundance analysis so we could see how similar they are between the different samples. We also did kind of correspondence analysis where you can include environmental parameters. And then we also did co-correlation. So who goes up and who goes down at the same time? Do they co-correlate either positively or negatively? So I'm going to distill this down and say that we use something called amplicon sequence variants. All you're going to say is those is going to be our indicator of a species. Okay, and actually it's a subspecies. Okay, but there's a variety of ways to do it, but we chose that method for this particular study. And so I'm going to refer to them as ASVs or taxons. And so they're basically who we identified. Okay. And so our very first question we wanted to ask ourselves is to continue with what Leonor talked about earlier. And does cropping system influence the abundance of fungi and FV in bulk soil? But when Leonor showed her data, that was actually at the end of the growing season in October when we did this. And so we did this during the times that we sampled during plant development, okay? And so our approach was quantitative PCR, but since we had so many samples, we also did a high throughput microfluidic approach. And then we looked at both total fungi using the 18S ribosomal RNA gene, as well as the intergenic spacer that Leonor talked about. And this is just bulk soil. We're gonna look at the 3 different cropping systems. On the top, we see here the bulk soil. And our color coordination is red's always going to be conventional, green is the 3-year diversified, and blue is our 4-year diversified. And our top is fungi, and this is FV in the bulk soil. And you can see, at least up to R2 in the bulk soil, we really weren't seeing much difference between the different cropping systems. Whereas, and same as with the total fungi, not much of a difference, okay? That was a bit surprising because given what we saw observed earlier, we would have thought there'd be other outcomes. However, when we look at what's on the rhizosphere, that's on the physically associated, the root and the rhizosphere soil, we saw some really different trends. First of all, if we look at the total fungi, as you go through your developmental stages that correspond to V2, excuse me, this would be V4, V1, V4, and R2 developmental stages. What we observed is the total fungi seemed pretty constant between all the different cropping systems, but by R2, we started seeing a divergence where the diversified system total fungal populations plummeted, okay? We also noticed that there was a slight decrease in the conventional system, but it was relatively mild. Okay, but really it was a big decrease in the 4-year diversified system. It paralleled what we observed with the FV. Okay, there's 2 points we want to make with the FV. First of all, at V4, you see the beginning of a decline in the diversified 4-year system. But you also know that the abundance of the FV in the conventional rhizosphere is much higher in red, a little lower in the 3-year rotation, and lower yet in the 4-year rotation, suggesting once again at that interface between where the fungus is going to interact with the plant, there's an influence of the cropping system on its abundance. Okay, then we said, okay, what's inside the plant? Okay, so that's where we look at the endosphere. Okay, and with the total fungi, you can see that there's no real difference across the different developmental stages. But early on, even though in the rhizosphere there's very similar abundance between the 3 and 4-year rotations, there are some slight differences in abundance between the 4, the 3, and the 2-year rotations right from the get-go. So events that are occurring very early are dictating some of the outcomes later on, because you don't observe SDS until about R2 at the earliest. Okay, so V1 is laying the groundwork for what we see later on. Okay, and there's no real change in FV abundance through all these developmental stages. Okay, so then the bigger question is, well, what are the other things going on in the total microbial community? And that's where we use the amplicon sequencing. And I'm not going to show a lot of it. I'm just going to give you some novellas, in many ways, of what we're going to see. This is just looking at a phylum level of who the different fungi may be in the different developmental stages in the rhizosphere and endosphere. I'm not going to worry about what's going on in the bulk soil, okay? And you can see that there's some differences, okay? And they're not dramatic differences because at the phylum level is a very poor level of discriminatory power among the different organisms there. You really have to look at more subspecies level or lower to discern what might be going on biologically. But we do observe some interesting trends. There's clearly a lot more Ascomycota relative to other organisms, but Ascomycota is just kind of like you lump everything into one group. It's taxonomically not well resolved. And we do also see some other different groups that increase the Basidiomycota increase in abundance. We also see some evidence for a high number of the Glomeromycota. Yes, the Glomeromycota early on, suggesting there might be some other interactions with important fungi. So this didn't tell us much, okay? And I could say the same as for the bacterial ones, and there's all the different groups you can look at. So let's look at really the Fusaria, which are the ones we're interested in. And so we look at the Fusaria family. So Fusaria is in a family called the Nectriaceae, and using our bioinformatics, we could actually break this family into 3 different genera. The green genre, unknown, okay, Marianae, okay, and then the Fusarium, of which includes the Fusarium FV, okay, and other Fusarium. And as you can see that there is some differences in relative abundance. It's not an absolute quantification. We're just saying of all the fungi there, this is the relative proportion of this group. And within this, we're looking at the total Nectriaceae, and we can see that they vary by cropping systems a little bit. Most notably, the unknown is most abundant in the 4-year systems, and the lowest abundance generally tends to be the Fusarium, okay, in the diversified cropping system. Particularly, as you can see, there's a real shift, particularly by R2 in the abundance by length of the rotation. So that gave us some information to suggest that the cropping system's influencing these particular groups of fungi. And so we want to do a little bit more different type analysis, and we want to do what's called to assess really what biological or environmental parameters are most predictive of how a cropping system would influence the community structure. Okay. And so we relied on something called canonical correspondence analysis. It's a constrained analysis that where the output includes significant variables as vectors. And the length of the vector tells you the length of the significance, and it points to the direction of usually the greatest increase, okay? And because of the type of analysis, we analyze the fungal communities and the bacterial communities separately. And to constrain it, okay, that's part of the name implies, we only included the parameters that we thought were most relevant. And what we did is we took bulk soil and we did all kinds of measurements on soil physical chemical parameters. We did not have enough rhizosphere soil to do those. Okay, so everything that looks at the rhizosphere is relating it to what's in the bulk soil just out of necessity. And then what we did include for each, the bulk soil or the rhizosphere or the endosphere is our measures of the FV populations, the fungi, the bacteria, and the archaea by quantitative PCR, okay? And including all the other microbiome data points, okay? Each of these samples has maybe 15,000 or 20,000 reads, which is like doing a census of going to Des Moines and saying, I'm going to get a census out of 15 or 20,000 people. Just a snapshot of 15 to 20,000 residents of Des Moines, which is poor. If I went to Ames, that would be better, okay? And this would be like us doing 15 to 20,000 going to Tokyo, okay, which is 25 million. Okay, that's just a sense of the scale differences. And we removed highly co-correlated parameters. Organic matter, really is equivalent to total carbon and total nitrogen. We just did one of those, okay? Organic matter. pH, same thing, pH, buffer pH, and bioavailable iron co-correlate. We only did one of those parameters. And we looked at them each individually. We did bulk soil, and then we did rhizosphere and endosphere. And so I'm going to first show you the bulk soil. And in the diversified system, you can see here the red, once again, is the conventional. The green is a 3-year, and the 4 is the blue. And the arrow shows the vector towards what is a significant variable. Okay, we're only including those that are significant. Okay, and that had to be at a 0.05 level or greater. Okay, or lower, I should say. And so what we see is that the fungal communities basically, Fe abundance goes here, pH is there, phosphorus, and you can explain some of these by the different environmental parameters we measured, okay? First of all, FV and organic matter vector in opposite directions, okay? That might be not surprising given conventional versus the diversified systems. The conventional fungal communities are also associated with ammonium, okay, which we didn't anticipate. Okay, so this led us to say, okay, this is just bulk soil. We were interested in trying to take this information and look at what the rhizosphere community as well, because we argued what you measured in the soil physical chemical properties of the soil would translate into the rhizosphere and endosphere. Okay, they're going to influence it. Ideally, we'd have enough rhizosphere soil to do all this, but we just couldn't get enough material. And the FV vector towards the 4-year system as well, suggesting that was interesting that it's kind of in between these two. It's kind of vectoring in between. So it's really not being explained by the conventional system or the diversified system. So it looks like it's going there, but it's really kind of in between it, okay? Which suggests to me, these physical chemical parameters were not very predictive of it, because we know where the incidence is. It's higher here, okay? So something else is driving the FV abundance. So what we also did this in the rhizosphere, okay? And so I'm separating out the fungal and the bacterial here. The other one was just for fungi. Okay, this is the fungal community. You can see a certain profile. Once again, for the bacterial, it's slightly different, but there are some similarities. In the rhizosphere, FV abundance vectors towards the conventional system, as well as you do right here if you're using a fungal community profile. And fungal abundance kind of sits there in the, once again, kind of in the middle between the diversified and the conventional systems, not directed towards anything in particular. And we also see some other trends, particularly organic matter again, and ammonium playing a particular significant correlations, okay? And then when we go to the endosphere, we really don't see much of a pattern of relevance, okay? And that's probably not surprising, okay? We're just too far removed from the soil. So the last thing I'm going to talk about is, well, microbial communities are really complex. There's a lot of different organisms that we're looking at. The number of, should we say, species is something like 5,000 to 6,000 that we surveyed inside each set of samples. Okay, that's a lot. And so one of the ways to look at them is you can look at them individually, what the relative abundance of each of those taxons is, or we can say, well, if this increases in abundance, does another species increase as well, or does it decrease? Or is there no relationship between the abundance of species X and the other myriad of other organisms there? And this is what we call, bring us into what we call co-correlation or co-occurrence analysis. And you can basically generate Spearman correlation coefficients or Pearson correlation coefficients with all of them. You have significant statistical criteria, either a correlation coefficient that is significant at greater than some level, the cutoff, okay? We usually use about 0.65 to 0.8 as our cutoff correlation coefficient, and it has to be statistically significant. But what we do, one additional thing, because that's a lot of, you can imagine the heat map of that 6,000 by 6,000 array. You can't really look at that. So what we do is we do a network analysis. And this is borrowing upon the physical scientists. This was first used actually to predict where electrons were in uranium, okay? And they actually turned out, Dyson Freeman got the Nobel Prize for this in part. And what we do is it explores the potential organization of interactions within the community, okay? And it can be used to predict putative keystone taxa, okay? So what it includes is a couple definitions. A node, or basically this would be any taxon that has a significant correlation with another organism, okay? If there's no significant correlation with anything, it's not in our network analysis. And we actually lose a large number, okay? A module is a group of nodes that are more highly associated with each other than they are with other groups, okay? And then the links are whether or not there's a positive or negative correlation between one or more taxon. So a taxon could have one positive link, or it could have 50, because it correlates in abundance with 50 other organisms, and they can be positive or negative correlations, okay? So we did this type of analysis, and we use something called the random matrix theory base, which allows us to see whether or not our network can be randomly generated. And we can't generate it randomly, which is good, okay? Suggesting there might be some relevance to it. And so when we look at this, and I'm only going to talk about the rhizosphere, and what we found is the microbial communities become more interconnected in the diversified rhizospheres over plant development. And what we found is that the number of nodes, if you look in the blue, increases at the R5 stage dramatically. There's some small increases in the number of nodes that are taxons in the other two systems, okay, but most dramatically in the Fourier diversified. But the modularity, that is the number of modules, okay, that correlate with each other, basically stays the same in the Fourier system, and there's some slight increases or dramatic increases in the 2 and the 3-year system, okay? The other piece of information that I'm going to use to describe this is the links. How many linkages are there between the different members in our modules? And there's a dramatic increase in the number of co-correlations. In other words, you increase the number of nodes, but you have very few modules. So the module has a lot more taxons, and they're highly interconnected with each other in the diversified system. In the conventional system, there's lots of individual modules, but they're not highly connected to each other or within a group, okay? Suggesting that they're like isolated, okay? And ecologically, we think that an ecosystem is more stable when it's more diversified and more highly interconnected. Suggesting the conventional system is basically facilitated development of communities that are more modularly and isolated and less connected with each other. And that's also when you look at our connectivity, which is basically our number of links to the number of modules, you can see this most precipitously. In the conventional system, it gets less and less interconnected in the conventional system. In the diversified Fourier system, there's a dramatic increase in the diversification and interconnectedness, okay? 3rd-year system, kind of in between, don't know really what's going on here, but I bring that up because this looks like the inverse of what we observe with the FV abundance. And so that allows us to speculate, is the decrease in fungi and FV due to competition that is occurring in these Fourier systems that's much greater than the other systems? And I'll also point out, almost always it's 99%, 97% positive interactions. Fourier system, it's actually only 80%. So there's a lot more clear negative interactions occurring here, which are giving us a little bit more ideas that there might be some negative competition that's facilitating this decline in the rhizosphere. And so I went through a lot of information, And I just I'll just do a quick summary on this. Fe abundance and the rise of industry decreases with rotation. That is something what it might explain clearly the disease observance is, it's just not as abundant in the rhizosphere. They're not getting in there. Even though they're in the soil, they just can't get through the rhizosphere, and then they can't get into the plant, particularly inside the plant, much lower numbers. Something keeps them from getting in. There were greater differences in community composition between conventional diversified systems than between the diversified system. So you go conventional to diversified, big differences between 3 and 4, a little smaller differences. In the endosphere, fusarium relative abundance is highest in the conventional system, which increases as the plant develops. That might reflect what we observe also, as well as other things that are going on in this system. And we also talk a little bit more about how there's the Fe abundance of bulk soil and rhizosphere is correlated with bacterial and fungal community composition. That's a canonical correspondence analysis, but aren't real predictive of what is going on. Okay, not like I thought I was going to. I was really disappointed. At first I was excited, and then I realized after thought about it, it wasn't what I thought it was going to be. And the diversified systems microbial community was explained by higher levels of soil organic matter and pH and ammonia. And we could have almost predicted that from what we already know about the systems. And then these interconnected communities over time. This isn't touched upon anything about the bacteria. And as Leonor said, there's years of data analysis and bioinformatics that we can still do. And so at that point, I would like to open it up to questions for either Leonor or myself. Yeah.
Is there any indication that the plant has any control over what's going on here? If you look at like the endosphere community, what's in the plant, does the plant have any control over what's going on, or is it just truly a passive process and the plant takes what it gets?
I'm going to say Leonor's data shows that obviously different genotypes have different influence susceptibilities. So the plant does have a control. Okay. In terms of the whole microbial community, yes and no. Plant genotypes do influence community composition, but a lot of it is environmental filtering. Whoever's there first gets in, has a greater chance of getting there. And if there's more of those in one habitat versus another, they might get there more readily. So Leonor's data with the field data clearly shows different genotype differences. That's definitely, once again, showing that plants can control who gets in.
One quick thing, if you don't mind repeating the question for those
yes,
when it comes from right here, and then if you don't mind standing a little closer to the computer if you're
Yeah, I think unfortunately for the microbiome data, we had a single variety at that point. Initially the trial had 2 varieties, and then eventually it became standardized and we had a single variety. And so we don't know how a particular variety, how it would differ in terms of, in the different systems. But yeah, comparing varieties for sure, there's some, at least on the pathogen side, you have less, usually a resistant variety has less pathogen Fe in the roots. And I mean, I'm sure it will affect other communities around in the rhizosphere just because of different compounds. But yeah, we don't have that variety by cropping system.
If they gave us the money to do it.
Maybe the next one.
Any other questions?
We have one online. Larry, I don't know if you can read it. It's fairly long. That might be easier for you to.
Okay.
If not, I can read it.
Okay. Larry, several
Larry?
questions.
Oh,
Okay.
okay.
This is from Michael Thompson. Okay. You lump pH with iron when doing the statistical analyses. Was that DTPA extractable iron? What was the correlation between the pH and iron? Since you found the microbial community composition was explained by pH, could it really have been iron instead? Boy, I hope I can remember all that. So the first thing about was that DPTA extractable. It was Malic 3. It was not DPTA. Okay. The other thing was the correlation. What was the correlation between the pH and iron? I don't recall. I know it was statistically significant. We only took those that were statistically significant. I don't know what the R squared was, but it was at least 0.05 or lower. Okay. Since you found the microbial community was explained by pH, could it really have been iron instead? Oh, I wish I could prove that. I really do believe iron is the driver of everything. Okay. pHs are not as important for bacteria as iron. Bacteria can adjust to the pH. It's whether or not they can get the iron. And Michael may know that I've had a soft spot for prairie potholes and the pH gradients there. And that has been something we've played around with. But I do know from the canonical correspondence analysis, you might have noticed that there was a few outlier dots. I can tell you which ones are the higher pH. Those were the ones. They correlate to a certain block that is wetter, higher pH.
They're close to that depression.
Yep, which usually the closed depressions usually have a little higher pH until you get into the closed depression.
There's another pH related question from Zach.
Oh, I don't know those values. The question is, what were the differences in pH between the systems? It really depends on which block you're in and which plot. I mean, there's at least 3 or 4 different soil series at the Marsden site. Most of it being Webster, which is 7.2, 7.3. There's some Harps Canisteo, which is where you get to the lower pH, the higher pH, and then some preponderance of Nicolette Clarion, which once again is going to get you a little lower. The pHs are slightly different, but I can't tell you which directions. Okay, and it really varies. It influences a lot where you're at in block 4. Because block 4 is very heterogeneous in pH.
I just looked it up. When we sampled a couple of years after you did, Larry. It was the 4-year system. We didn't do the 3-year. It was an average 6.8, and that was across all blocks. But you're right, there was a lot of variability across the blocks. And the average of the 2-year system was lower at like 6.3.
Yeah, in our maize study, we did a similar type of thing with maize 1 year, 2 and the 4, and we could go and pick out where those samples came from by their way they're outliers relative to all the others. pH is associated with, or like soil series is associated with these differences in community structures. It's just, the problem is heterogeneity. It's really hard to have enough replication to really pull things together when you have even a soil series. Soil series is a very crude measurement, maybe on a microbial scale. Because microbial scales are microns.
I've got another question, if you don't mind.
Mm-hmm.
I know we're running low on time. Could you pull up the canonical correspondence analysis? I'm not familiar with those. I've done principal components analysis, and usually, you just like you did with the CCA, you had the percent, and I assume that's percent of explained.
Yeah.
And that seems like a really low, and I think this is what you said too. You said these environmental variables did not explain the differences in these communities at all. And that seems like a very low score, at least in a PCA it is.
It is. It is. In fact, compared to a PCA, because it's really not that different from a PCA, except for include these other constrained variables. If you did a redundancy analysis, which is a different form of constrained analysis, you get a little better, larger values. I wouldn't say they were dramatically different, but you had poor resolution. Everything's kind of lumped together because of the way it looked. These are the most visual. We basically came to the same conclusion regardless of what form of analysis we did here, but this one is easier to visualize. Okay, that's one of the reasons we showed it, and that's probably why we'll use this one in a publication, not at least in the primary component. You can do the same type of principal component analysis without including all the different environmental factors, okay? And just do the microbial those what we call the beta diversity. So we're taking out how all the different taxons in that one sample, and we're putting it, that's one dot right here. That reflects all 6,000 species or whatever it is, okay? Put in distilling into one dimensional space. When we do that, actually, they're more tightly clustered. So the small amount of variation from these different environmental factors, coordinateness, does help separate it a little bit, okay? And actually, I think it's you can see the differences between the cropping systems much more readily here, but the ideal way is to look at this in X dimensions of space. And that's where R is not good at 3-dimensional plotting. And sometimes we will download all this data and put it into a graphics package so we can spin the axes. So you can actually see them get better resolution. But here, I don't think it'd make much difference. I mean, if I put in the third coordinate, it'd be less than 1.6. You know, but when you're dealing with 20 or 30%, the third variable can actually help separate something that might look on top of each other, but they're actually, well, actually they look like together, but they're actually on top of each other, separated.
That was a good explanation of these plots. It took me years to figure out how to do these.
I'm not sure I really still know.
Nothing else has come online. Any other questions from here?
Well, I want to thank everyone for allowing us to come. I want to thank Leonor for convincing me to do this. I originally said no to Marshall, but I said if I could only do half a seminar, then I'd do it.
It was fun doing a double header. Thanks, guys.
Mm-hmm.